Hi, Xenia.
Hi.
Thanks for coming. We'll wait until it's time, like we wait 5 minutes and give people time to arrive, and then I'll introduce you.
Thank you, Katerina. Nice to hear you today,
Yeah,
this evening.
nice to hear you too. Hi, everyone. Thank you for coming. Um, we're waiting a few minutes. Your audio is working just fine. We were waiting for people to arrive, and yeah, so we can slowly start the room. Please, everyone that wants to come up to the stage, raise your hand, and you will be able to ask your questions. But first, let me introduce you to Dr. Ksenia Krasileva. We are very honored to have her today. She will be talking about her latest research, which I think is really groundbreaking and very interesting. So thank you so much. And let me give you some background information about Dr. Krasileva. Um, she is an assistant professor at the University of California, Berkeley. She is an interdisciplinary scientist who studies genomics and plant immunity. Krasileva holds both a bachelor in science and a PhD degree from Berkeley, where she studied plant microbe interactions, and she was trained in genomic and computational biology. She did her postdoctoral work in wheat genomics with George Dubokovsky at University of California, Davis, supported by USDA NIFA postdoctoral fellowship. Krasileva received the Carlotta Award for contribution to wheat genomics, Gordon and Betty Moore Foundation Inventor Fellow, and NIH Director's New Innovator Awards for new innovative combining evolutionary and computational biology. The Krasileva Lab at Berkeley maintains research interests in the biology of genomes, evolution and function of innate immune systems, and of plant pathogen interactions. You can read more at her lab website, which is krasilevalab.org. I can post it later, the link to her lab, and you can also find her on Twitter. Thank you so much for coming, and we are very honored to have you, and we are very excited to hear about your research. The stage is yours.
Thank you, Katarina. It's lovely to be here and to present the story behind the paper to you. I'll start by saying that I'm really speaking with a heavy heart today because, as many of you can guess from my last name, I was actually born in Russia, in St. Petersburg, and what I'm seeing today is really worrisome to me. And my heart is with the people in Ukraine as well as with the protesters right now in my hometown and many towns across Russia. But what I'm going to tell you today is a good story, a story how the computational biology and the main breakthroughs that are happening right now.
Ksenia, can
Yeah,
I? Yeah, I wanted to. I wasn't sure what to do today. If I should put some links to support for humanitarian and medical support for people in the conflict zone. So I thank you that you mentioned it. I didn't want to take the stage away from you. So please go on this link tree if you want to donate, if you want to have more resources. I got this from a room that was led by people from NPR, the National Public Radio, earlier. So yeah, go on there to have some reliable resources and also links for donations that were verified by trustworthy people, as far as I can tell. Yeah, I'll switch in a second the link back to Dr. Krasileva's paper.
thank you, Katerina. I really appreciate that. I wrote a letter to my colleagues today, and I spoke to my lab today, and I just urge you all to just look at the events, but also keep an eye on the protests that are going on and the brave people who are protesting this war. Um, okay, back to computational biology. So, what I believe is that we have a lot of advancements in the technology right now, including computational biology. And now is there really a time to combine it and use it for the good to solve problems that we couldn't solve before on a completely different level? And one of these problems, was for us has been how does a pathogen becomes a pathogen? And we understand it better maybe with viruses which have only a few genes, and again, it's hard to study them as you probably know, but they have only a few genes and they're successful, very successful in causing infections. Bacteria might have, several dozens of genes that are secreted into the host, and they're also really good at causing the infection. But if it comes to the fungal pathogens, there isn't really a minimum number of genes they need to cause an infection, and they can secrete literally hundreds and thousands of proteins and molecules. And to decipher which ones of them are important for pathogenesis, which ones of them can we target, how are they all related to each other, what makes a fungal pathogen a pathogen, has been an unprecedentedly hard task. And we argue that in our paper that computational structural genomics is one of the approaches today that can allow us to address that question. Some of you might not have been thinking about a fungal pathogen. What does a fungal pathogen looks like, right? It is a problem for human health. We have several major fungal pathogens of humans, but we don't normally see it in our everyday life. Now, what I want you to think about when you think about fungal pathogens, think about the animation video of Japanese Studio Ghibli called Nausicaa. And if you've seen Nausicaa, you've seen this post-apocalyptic world that's inhabited, that's been taken over by a toxic fungus that releases its spores in the environment. Now, that is actually not far away from the reality when it comes to the fungal infection of our plants and especially crops. Fungi are really, really good at infecting plants. They're really, really good at infecting huge areas that are planted by crops such as wheat or soybean or any other major crops out there in the world. And they release clouds of spores in the environment that they can be then transported over huge distances through wind, by air. Those spores are about the size of a pollen grains, and so all of us during those seasons, we inhale them, we inhale those spores. If I go to a wheat field on some of our trials, my shoes turn orange from the spores that are present in that field. And the current way of controlling these fungal pathogens is fungicides, is chemicals that are being spread maybe 3 to 5 times a year. They're spread by airplanes, they're applied to most of our crops, and fungi can also become resistant to the fungicide. Now, what my lab studies and the paradox that we want to address is how to utilize the natural native immune systems of the plants and of all the organisms that do not have adaptive immunity, antibody-based immunity, to defend these pathogens. And the plants have fantastic repertoires of defenses, and in nature they're capable of recognizing rapidly evolving pathogens in their molecules. And so what we keep thinking is, how do we identify these virulence factors in the pathogens, and how do we identify maybe what's conserved across different pathogens, and how do we target them to boost immunity of our crops? Now, as I mentioned, in a typical plant pathogen, a fungal pathogen can encode between 500 and 3,000 secreted proteins that it will release in the environment and inject into their hosts. And these 500 to 3,000 proteins, often they don't look like anything else. They don't look like each other even if you just take their primary amino acid sequence. And sometimes that's because they're rapidly evolving. So the sequence similarities degrades over time too rapidly. Sometimes that is because they're highly adaptable and they arise quite rapidly in the course of a pathogen evolution or fungal evolution. And we were quite at loss, when I was doing my PhD, I did it on one omycete or molecule, one virulence molecule at a time. And typically, one PhD student, one virulence molecule. It takes 5 years to study it. If you have 2,000 of them in one pathogen, that's incomprehensible. How do we cover them all? And what happened in the last couple of years, we had this breakthrough in machine learning approaches' ability to predict three-dimensional structures of the proteins. The approaches were developed first, they were developed in academia, and they've been developed for the past 30 years by the labs, such as David Baker's lab, and they've been also picked up and started to be developed by industry, specifically by DeepMind company that released AlphaGo. They defeated computer in the game of Go using machine learning, and the next problem they attacked was making these models, predicting structural models of proteins. And in my lab, a PhD student pioneered this work. His name is Kunyang Seong. He's a Korean American, and he came to me during his first year of his PhD and said, Ksenia, there has been this breakthrough. I want to apply it to resolve three-dimensional structures of thousands of fungal pathogen molecules. And I want to be able to compare these proteins in the 3D space to see what are the similarities and what are the differences. And these tasks, although the machine learning technology was available, was very, very, very difficult, because it turned out it was quite difficult to make sure everything runs. It takes immense amount of computational resources, but Kunyang persevered and he made it work. And what you see in this paper is the result of his first study. And it was the first study in the field that applied this computational structural genomics approach to literally thousands of molecules. What we found was very interesting. We found that the modeling, the machine learning modeling, in fact, does work really, really well. And it is able to predict vast majority, give us high confidence structures for vast majority of the protein. It didn't work on everything. So we also identified groups of proteins that couldn't be resolved by this method. Some of them there might be intrinsically unstructured. Others might be lacking enough information in the data sets available today to reliably predict the structures. And for those proteins where we did identify the structures, it was reliably, that was around 1,000 proteins for these fungal pathogens. Kunyang was able to compare them in a 3D space and was able to form similarity groups. So sequences that look nothing alike on their primary amino acid level, all of a sudden through that lens of 3D structural genomics, we saw how they looked similarly. And so we were able to classify these thousands of proteins in about 100 structural groups. And we were able to identify in this specific pathogen specific structural groups that are expanded, that are, they're unique to this pathogen. We were also able to see secreted proteins that look similar between this pathogen and some of the other pathogens for which structures were available, and formulate a couple of ideas. One, we think that we can apply computational structural genomics to the problem of resolving virulence factors secreted proteins and identifying structural groups. So we can start to do evolutionary analysis in the 3D space and look how do the actual shapes of the proteins change. Secondly, we observed that seemingly unrelated pathogens utilize similar structures, similar folds. So we can start to ask questions of what does distinguish a fungus that's a pathogen from a fungus that just leaves, that are beneficial to a plant or that lives saprophytically in the environment. And then, well, can we target the specific classes of proteins that are unique to pathogens? The other question that we could ask is, where do these pathogen factors come from? And currently, in current work that Kunyang is continuing, we hypothesize that they actually are derived from maybe a benign secreted protein that are normally a fungus utilize it to survive in the environment, and they're then expanded in copy number and adapted to infect a specific host. The other key question that we asked is, it's often hard to predict a function of these secreted proteins, because they don't look like anything else in the databases. And by using the structural 3D information, we were able to say, hey, this protein looks like an enzyme. It looks like it might be chewing something, and the other protein might look like another. So we can also do functional annotation and get a better idea what type of activities these proteins encode. Now, where does it lead us next? And I really want to, spend more time discussing it and hearing from you. I think it leads us next to a very exciting era where we can do not only computational structural genomics, but comparative computational structural genomics. And so what we're doing right now, we are applying this process, and I would urge anyone, if you're working on a pathogen, apply computational structural genomics to your pathogen of interest. We're applying this process to around 40 different fungi, including those that just live in the environment, a couple of mushrooms, and many plant pathogens. And we're comparing what type of structural groups distinguish different pathogens, and what type of structural groups might be associated with their adaptation to particular hosts, such as wheat or rice. What type of structures might be associated when a pathogen jumps the host, and what type of structural groups are associated with the pathogen's lifestyle, whether it's prefers to live on a live plant tissue or it chews up a dead plant tissue. And overall, very exciting area of research. I'm quite proud of Kunyang, and I'm really happy to be involved in this area. And I would love to hear more from you and be able to answer your questions about it.
Yeah, thank you so much for that great introduction and giving us an overview of your amazing, very interesting work. So, yeah, please go ahead. If you have questions, flash your mic or, go ahead, please. Well, if nobody asks a question, I will go ahead and ask a question. So, I think your research is so do you see that your research is interesting, especially for agriculture and the future, for creating basically a more sustainable agriculture? Since if we can better know how pathogens basically are destroying crops and so on, can we develop a less harsh for the environment, a less harsh pesticide version based on your work?
Yes, absolutely, Katarina. And what we're thinking about now, and we're thinking about the application of this work. We're thinking, we can start to develop receptors, immune receptors against these molecules, but we need to be able to pick, right? Which molecule do we choose? And this structural genomics work allows us to get structural information, but B, get information of what is conserved, right? What might be indispensable for the pathogen and target that. And I think it's an exciting area right now because the synthetic biology and ability to synthesize DNA and to synthesize what we call designer receptors would allow us to essentially develop immunotherapy-like approaches for plants, for agriculture, and say, hey, we can now give plants an ability to recognize a particular pathogen rapid in response, and it's specifically important for crops. In natural pathogen systems and natural environment, a field of grass, you can rarely see it completely infected and dead, right? Because on a population level, plants on a level of a field of grass, plants are capable of generating enough diversity in these immune receptors to recognize pathogens. But what we've done in our crops, we plant monocultures. We plant the exact genetic variety that suits us, that tastes best, and we lost a lot of that immune diversity, and we need to bring it back, and that's what we are working on as well. So looking at the pathogen is like finding what's the Achilles heel that we can target.
Yeah, that's such important work. That's amazing. Another field, since we are here on Clubhouse, is what I'm thinking about is for terraforming other planets. I think your research would also be quite helpful for that. Have you thought about that, or have people approached you for that type of field?
Can you explain more, a bit
Yeah,
more?
let's say, we go to Mars or try to live on the moon. It's always helpful to know the Achilles of plants for that very
Right,
fragile
right.
condition.
So, yeah, well, I mean, don't bring plant pathogens to the moon. That would be my main advice. But the fact is, if we do, they're likely to win unless you have, something like innate, immune receptors in a test tube with you to combat them. So definitely in any type of truly, sustainable world, we do need these immunity genes for plants to be ready, and we need the technologies that we currently do have for human health. We need it to be available for the health of our plants and for the health of our planet. And we have all the knowledge and all of the technologies to make this breakthrough. It is still a very much an intellectual and challenge, and that's what we're addressing. But absolutely, you want to be able to make a vaccine for human diseases. You want to be able to make vaccine for your plants as well, especially if you go into space.
Yeah, go ahead.
Yes, I have a question. Uh, so I just quickly read the abstract. I think this is very interesting proposal that I mean research findings that but as a layperson, I would just like to understand the what is the achievement here. The seems to me is a almost like a as not an expert in this field, I would describe this is almost like a from bottom up versus top down methods. You're looking at the effector, the molecules, and try to almost reverse engineer what the protein, and then go up instead of top down, like you from the gene, the what do you call it? Also, if you can elaborate a little bit more about the top-down method, the homologous type of approach. Yeah, that I would appreciate. Yeah, thanks.
Of course, Frank, and that's a great question. So, what happened about 10, 20 years ago? We had a revolution in genome sequencing and our ability to decode genomes, and that gave us information about the DNA that's present in almost any organisms. And we've now moving towards the era where we'll have these sequences for almost every organism on Earth. There are projects like that. Now, this gives us the DNA, the genome. The DNA uncodes genes that are then transcribed into RNA and translated into proteins. And what builds up cells, right, and what builds up most of the functions in the cells is the proteins. And although we can now tell, the amino acid sequence of these proteins, what their function is encoded in the 3D shape of the proteins. And in order to know what the proteins do, we need to know the 3D shape. Previously, and experimentally, the structures, the 3D shapes of the proteins are available for less than 0.1% of genes overall. In a fungal pathogens like Magnaporthe, those structures are available for maybe 10 or 15 proteins, like experimentally solved structures out of thousands and thousands. And okay, so we have this vast amount of DNA information. We have this absolutely minimal amount of what these proteins really look like in 3D space. And all the previous methods, what they relied on homology modeling, meaning for this 0.1% of proteins where we have experimental structure, if we can find, if our sequence looks something like that sequence for which we know the structure, we can say, hey, maybe the proteins that we are decoding in our organism, they look something like what's been experimentally shown. Now, when it comes to fungal pathogens, there's no detectable homology. There is no thing that looks like these secreted proteins. They evolve too rapidly. They change their genomes and amino acid sequences. The genomes encode too rapidly. So we cannot reliably use homology methods. We don't have any point of reference. We didn't know what they look like. And this machine learning approach, what it utilized vast amount of information, sequence information, to say, hey, if a particular points in the protein amino acid residues are co-evolving, that a change in one leads to a change in the other, we can start use this information to de novo predict folding of that protein, the shape of the protein. And that's what we applied. That's the breakthrough that we applied here. But basically what we are after is, what do these proteins look like in 3D in order to understand how they function? What do they actually do? I hope that makes sense.
So you're using machine learning algorithm that would require identify some features. What are the features you're looking for?
Yes.
Sorry.
So the machine learning algorithms today that solve the protein folding structures, they've been trained on two things. They've been trained on the existing databases of solved structures, plus the features are the co-evolving amino acid residues across many proteins. So they've been trained on very huge data sets and on the data points within the proteins that change, that codependent on each other. And the idea that these algorithms used was that the points that are codependent, that change together, they're more likely to be closer in 3D space. And the breakthrough in these machine learning algorithms, they've been shown to outcompete homology-based modeling in 2018 in a community-wide competition that happens every 2 years. And that's when they caught our eye, and we decided, well, because they work well enough de novo solve protein structures, we can now apply it on a genome-wide level to resolve thousands of structures of thousands of proteins and be able to compare them in this 3D space.
I see. So just my guess is that the previous homologous methods, they had their metric of the definition of similarity is less granular. than the feature that you're using. You're breaking it down to finer components, and starting from there, and redefine a similarity metric and regrouping. Can I? Am I right on this line, or
Um,
I'm confused?
mhm. So the previous homology-based modeling required the presence of a homologue, and a homologue is a sequence with enough sequence similarity to say that your sequence likely is related and evolved from a common ancestor. So it requires a homologue. In the secreted with these virulence factors of many pathogens, there are no detectable homologues, especially with salt structures. So they were blind spots for this method completely. It's not like the structure could be predicted badly. There's nothing, because you put it into database and you get nothing. You get nothing that looks like these sequences. They're diverging too rapidly. The machine learning methods, they were trained on large data sets where there are some homologues, but they were then, we could apply them to de novo structure prediction, to the folding using primary sequence alone without having a homologue with a salt structure. I hope that makes sense.
I think I got it. Yeah, thanks. I think the key is the uh, innovation. Uh, you mean, uh, yeah, getting beyond the homologs. Yeah, now I got the point. Yeah,
Exactly,
thanks a lot. Yeah.
exactly. And that's
Um,
particularly key to the pathogens.
may I ask a
Sure.
question, please? Um, well, thanks. Actually, thank you for the introduction and for the fascinating work. And just in a brief, I got two questions. So, by what extent could we address just from the structure of the protein, the functional proximity of it? I mean, whether it could be enzyme or something like for what percentage do we can get the function based on the structure or of itself. And my second question, could we extend the same pattern for from just the structure or the single sequence of the gene itself or the structure of the protein to get a clue about the phenotypic or morphological uh, expression or genomorphic expression? Thank
Sure. I think the first question for me is the answer is easier to that. What we do when we make the structures, we group the structures into similarity groups, right? Saying, hey, this is a group of proteins that looks alike in 3D. And then we're able to compare it to the 3D structural databases of anything and everything that's available. And that often does give us a clue what the protein might be doing, or what are other proteins that have been characterized it looks like. Um, it does give us a good a starting point. It doesn't tell us for sure, but it's a kind of a hypothesis generation point. It gave us a hypothesis generation point for many of these proteins. There is still a remaining, what we call novel folds that haven't been seen before. And for these, we don't know, right? They don't look like anything else. They're exciting as well, because they might be something that pathogens really use, but using the comparative structural genomics, I think we can also look at this prevalence of this novel folds as well across other pathogens. Now, your second question was, can we predict what's essential for a pathogen? And I think here the it's harder, right? A pathogen is really, really good at its job, especially a fungal pathogen that can rely on thousands of proteins. There's often redundancy, meaning, well, if you can lose one gene and use the other one that does a similar thing, our ability to make the structural groups gives us a better idea of what's redundant. And for example, now with the genome editing and CRISPR, we can look and test what particular structural group might be essential for a pathogen, because we know which groups of gene now to target, to pick, edit, and see if there is a phenotypic effect. And before that, we would have been just shooting in the dark. We didn't know what groups with what.
Thanks a lot. It was very informative.
Can I ask a quick question?
Yes, go ahead.
Professor, what are the next steps that you and your team are gonna do? Because I was reading the end of your paper, and it seems that the rest that you haven't identified, I guess they are related to pseudo enzymes somehow. And my second question would be, what exactly are pseudo enzymes? I'm not too familiar with that term.
That's a great question. So the next two things we're doing, and right now, is this comparative computational structural genomics, where we're applying that exact same approach to a bunch of different fungi with different lifestyles to see what's common across them and what's unique to particular pathogens. And the second one is the pseudo enzymes are particularly interesting. And what we're looking at is, where do pathogen specific molecules come from, right? It's not like they come from out of space, they come from probably existing genes. And what we've observing right now is, sometimes there's a gene, an enzyme that might encode a very routine function for a fungus in general, just living in peace. But it might be utilized by a pathogen to evolve a new function. And what we see in the pseudoenzyme families is that the original enzymatic function is lost, yet there is a diversification of particular protein surfaces that we think might be repurposed to target some of the host proteins. And that's what we're tracking. Pseudoenzyme means it doesn't have, it has an overall shape of a known enzyme, but it's lost some of the residues that other people predicted would be important for the catalytic activity of that enzyme. And yet those are not pseudogenes, like they're conserved, they're expanding, and we see evidence of evolution acting on parts of them that might not be the exact catalytic site.
Thank you for your answer. My next question would be, how long did this whole study take you and your team to develop and carry out?
That's an excellent question. We started it quite early, and before the major press releases and availability of major algorithms. It took us about 6 months to run the analysis. It takes months to run the analysis, few months, and then and homology-based modeling, to which we were comparing, actually took longer. And then it took us another, and by us I mean actually Kunyang. It's led by one person and one absolutely brilliant graduate student with lots of perseverance and grit. It took them a few more months to analyze the data, but the actual lag time for us was to publish it. We were early enough with this data where we put it out and we pre-printed it, and it actually wasn't obvious to the communities that this worked well. So we had to talk to a lot of people. We have to explain exactly what we did, what controls we run. And so it took us another 6 months to publish it. So overall, year and a half from conception to publication.
And how long do you think it's gonna take you carry out to carry out the next few steps that you're thinking of doing?
Actually, much faster. So the analysis of the 40, so we scaled up, right? Analysis of the 40 pathogens and their secreted structural data sets are complete already. And right now, Kunyang is just doing the comparative analysis and writing the paper. And I predict that it would actually be easier and faster to publish the second study now that actually the community is much more comfortable with the ideas of machine learning to predict structures and the computational structural genomics approaches.
Yeah, hey, thank you, Katerina. And I just had a quick question. I don't have a biological background, but I do understand computers, and I just wanted to know. So your data model that you have been referring to, the AI model that's working right now, what has that been trained on? As in, has it been trained on the fungal pathogens from the human infections, or has it been trained on from the plant infections, or has it been trained on the animal infections? And the follow-up question for that will be, how long has been, or how old is the data model that you've been working with? Like, how long has it been trained for?
Yeah, so that's a great question. So the training is done by the, it's not done by us. It takes immense computational power and a lot of GPU power essentially. And the algorithms we're using right now, they're called AlphaFold 2. They've been trained, they developed by DeepMind and Google. They've been trained at Google on everything. So not just plant pathogens, not just human pathogens, on every single available protein structure. Because you need, as I said, the actual experimentally solved data set is absolutely minuscule. So you need to scrape basically everything and anything that's available. The training should be like, we're working with absolutely latest algorithms, and I'm not sure exactly when the training was updated for AlphaFold 2. I would love to have more computational power for my group than we have now and be able to train some of these algorithms ourselves and see how it affects the outcome.
Yeah, thank you for that. Just like last quick question. So you said like the successor from the program where they created AlphaGo.
Yeah, exactly. So for the machine learning methods for protein structure prediction, there are two main groups that reported the breakthrough in 2018. One was the academic group, a couple of academic groups, and they developed a program called TrRosetta, and they've been working on it for the past 30 years. And the other group was DeepMind, the same developers that developed AlphaGo. They developed AlphaFold. And both of these in 2018 outcompeted homology-based modeling and performed quite equally. And then in the next 2 years, DeepMind developed AlphaFold 2. And what they did several innovations. Some of them still remain a black box to us. Others are more or less clear. And the AlphaFold 2 now does outperform, including in our hands, the precision of structure prediction. It doesn't resolve many more structures. So about, if we take full secreted proteins, about 2,000 of them from this pathogen, both approaches, academic and the industry, they predict confidently about 60% of them. But AlphaFold 2 gives a better precision, more like atomic level resolution of the structures for which a good model can be obtained.
Okay, thank you very much.
Can I ask another question?
I think Abyss is flashing his mic, and then we can come back to you.
Okay.
Thanks, Garrett. Thanks, Ksenia. I've been listening to the presentation, although I have to admit that I came in late. So I do have a couple of questions. So the first one is that your I'm assuming that you're actually using the sort of like the data that's been sort of used by AlphaFold. I guess like when you answered Flame's question, I was kind of getting my reply at the same time. But were these structures? I guess my question is like, were these structures developed um, or at least like was there like X-ray crystallography actually done on those protein geometries, or was there any kind of novel technique that was applied to in order to determine the structure of the proteins for you to use as your training data, or at least like the data that you were interested in sort of predicting the um, the potential proteins that you would expect in pathogenic fungi? That's my first question. My second one is that um, we all know that but we do have bacterial infections, viral infections, for which we have most sort of like medications or some kind of interventions fully developed at this point. But I heard that fungal infections are rather persistent. I know that may not be your area of expertise from perspective that you're studying fungal pathogens with regards to plants, but um, um, what sort of effort is out there to kind of mitigate those persistent fungal infections that can actually endure the body temperature, which I think is the most one of the things that actually protects us from fungi to essentially um, have lasting infection. So those are my two questions.
Those are great questions, Abis. So to address the first one, experimental kind of validation, experimental data, there are about 20 or 30 data points available for fungal effectors overall. We did use them to see how good the predicted confidence scores corresponded to these experimental data points, to real ones, including we did look at specifically at the structures that had been solved after the models have been trained. Because again, the models, the machine learning models, they've been trained on, not by us, they've been trained, like we knew the data, but we knew the date when they've been trained. And so we were able to assess that. And what we saw is that the predictive confidence scores corresponded really well to the actual precision. And so we could use on those thousands of models, we can use the predicted confidence scores as a measure of the accuracy. And indeed, we used that, and we used a 0.5 cutoff that corresponds. Well, the program got the overall fold of the protein correctly. Specific atoms might be not in place. But we also said, well, if it's below that cutoff that we knew has a good value, then we're not going to do the analysis on these proteins. Those are not good models, etc. The other point that you're making is, yes, fungal, and that was, I was doing introduction, different pathogens can be successful with a completely drastically orders of magnitude different number of proteins they use. The viruses only, encodes a few genes, bacteria, a couple of dozens, fungi, many, many, many. And the infections are persistent. They're really good at doing their job. It's really hard to get rid of them. And that's one of our questions is, well, how? What do their weapons and secreted proteins look like? How can we target them?
Thank you.
I think Arvin, go ahead. You had the question, I think.
Yeah. So I was just reading the abstract, and then it says in that, if I can quote, it says, along with sensitive homology search, that you employed structure-based clustering, defining not only homologous groups with divergent members, but also sequence unrelated but structurally analogous groups. So I'm guessing, like, okay, the machine learning does its job and it's able to identify this, but have you guys postulated a mechanism as to how these are actually how these are actually related, and uh, why, I guess, some kind of causal mechanism for why they fold the way they do. Any more insight into that? Because I think from that sentence, it's you have genetically homologous proteins with different structures, but also different sequences with similar structures. So, is there any kind of deeper insight or some other level of clustering that you postulated or hypothesized about?
Yes, so Arvind, this is an excellent point. So the first point, you can have sequences that are drastically different, yet they fold the same. Um, and that is because the sequences in the pathogen, they revolve really, really rapidly. So they lose the similarity sequence signal. It's hard to tell that they are homologous when in fact they are. They're just diverging very rapidly. And they're still able to preserve the same structure because um, you need only actually a few amino acids to hold the structure together. And most of the amino acids that are on the surface of the structure can evolve away and then not without affecting the core, kind of the structural core. The amino acids that are pointing inside, um, that's what's holding the structural core together. Now, just by looking at sequence in string space, you cannot tell which amino acids would be pointing to the core, which ones are pointing outside, so it's hard to detect this conservation, that homology. The other point is, how can we have structures from seemingly unrelated pathogen, including a bacteria or plus a fungus, that look the same? Are these analogous, meaning that they were not homologs, they were derived independently? Have they been horizontally transferred, for example, between pathogens? Do they represent some very, very ancient, very, very divergent sequence? So that we cannot answer in this study, but that's why we're doing this comparative study, where we're starting to do comparative evolutionary analysis in 3D space. Hope that helps.
Yeah, if I can just dwell a little deeper into the one with the structure. So, if we're talking about, say, the amino acids that you're saying are um, sort of core members or sort of skeletal amino acids, and then everything else can kind of change around that. Have you been able? I mean, have you been able to identify the in the ones that you've run? Have you been able to identify the point at which these amino acids in the string, so in the one-dimensional string of amino acids that represents that protein, would you be able to identify that these amino acids have to occur at a certain location? And because I'm still trying to imagine, like beads on a string. If I place amino acid after amino acid to create a protein, which is a necklace, I guess. If I place 4 or 5, and then it turns out that every fifth amino acid is critical, then we can identify that because maybe it folds over with a radius of 4 or 5, and then those are bonded through hydrogen bonding or something. I'm not certain of what the mechanism would be, but I would have thought that the number would be conserved. So the spacing in the one dimension would be conserved for the critical amino acids. Is that wrong for some reason?
Arun, that's a great question. I can tell you that. So I have, when I did a PhD, we did solve like one structure of one protein. My whole PhD is 6 years, and another group, spent another 6 years, and they solved another structure, and our structures from two different pathogens, they look the same. And so that's where my interest as well started with that. What we can do and what we were looking for, exactly what you say, is like, well, what is the same in them? Like, what are these motifs? And the way one does that, we can make structure on structure alignments. So align structure, overimpose them over each other, and from that overimposing them, derive the primary sequence alignment of who aligns with whom in a primary space, and then specifically look of what are the amino acids pointing in, holding the structure together, and how do they align, what might be conserved. Now, even when we did that with the experimentally solved structures that looked alike, we were able to do that. We were able to derive this alignment, and we came up with 3 amino acids. So 3 bids on the string, W, Y, and there was one more that was slightly variable. And those are the ones that were conserved in holding the structure together. So the number of amino acids that are needed to maybe hold that together, it's not much. It's not a lot, and therefore it's it doesn't give much. It gives some information. We still call this structures WY, after these 2 amino acid type structures. But if you have, 300 beads on the string and only 2 of them tell you what holds this together, it's then hard to utilize this information in the primary sequence space to then, go and look for anything else that looks like that. We still did it with the reliable success utilizing hidden Markov models. But now I'm convinced that doing these comparisons in 3D space is much, much more powerful. But, Kunyang is actually thinking along that line as well. Can we now identify some motifs that are associated with particular folds? I don't have the full answer yet to that.
Yeah, that's awesome. And a last comment on that. So would those key amino acids then naturally be targets for some therapy? So for some pharmacology, if these things are pathogens, because then you change the entire structure pretty significantly, and yeah, probably the protein loses its function.
Oh, that's a fantastic idea. I haven't thought about it that way. On disrupting these particular critical points that holds the structure to either with, even if you send like genome editing or some, that's a fantastic idea. I actually haven't thought about it in this way. But yeah, that's a great idea.
Cool. Glad I could put that into your head. Thank you.
Yeah, thank you so much for all those great questions. Does anyone have another question? I think we have maybe 3 to 4 more minutes, if I'm not mistaken, Dr. Krasileva.
Is there any way to contact you in order to, I guess, see how the study is going, or to talk to you more about your other research?
Carly, that's a great question. Thank you for asking. Yes, the best ways you can contact me on Twitter. So I put my handle and my bio profile. You can look at my website and send me an email. And I can also, I'm always happy to correspond by email or schedule a Zoom talk. I have heard already from colleagues working on human pathogens, fungal pathogens. They love this approach, would love to apply it. And in general, I'm always happy to talk. So email or Twitter are both really, really good. And to follow me on Twitter, and I will tweet all of the research coming out of the lab. That's my professional Twitter account.
Yeah, or maybe you you could also come back here, maybe when you have updates at some point. This is such a great and informative room, so we really appreciate it.
Of course. Thank you, Katarina, for exposing me to this environment. It was new to me, and I'm actually quite eager to tell about it to my lab as well. And hopefully you can invite some of them here as well, and listen to all of the rooms and chats, and be part of this conversation.
Wow, yeah, that's wonderful. We would be happy to have your colleagues and your lab members in our club. That would be wonderful. And yes, come back anytime. We have amazing guest speakers. Of course, your work is quite exceptional, but we will have more guest speakers. Just for everyone, please follow the club Science Society if you click on the reminders of the rooms, you can, you will see when we have rooms like this. You can click on the calendar and follow. We also have a website. It's also on the bio of Science Society, where we have the upcoming rooms. Just to give you a quick overview of the few next rooms that are coming up tomorrow. We will talk again about innovations in renewable energy to address climate change. That's a follow-up room from a previous room. We have a philosophy of AI, building the minds of tomorrow. We have Khurram Zuberi. He will talk about his thesis that he wrote about this. And and then next week we have Dr. Mohanta explaining promising depression therapy she's been working on, and Dr. Weigel and Dr. Monroe. They will present this really groundbreaking research that DNA mutations are not random after all. So it kind of challenges the so far evolutionary hypothesis we have so far that mutations are random. So this will be a very interesting talk, and I'm very excited to have them here also. And maybe Dr. Krasileva, it's kind of in your field, a little bit in your field of research. Maybe you want to come and listen to them, but it's at 1 p.m. EST since they are from Germany. So I'm not sure if that's a time that you're available. But yeah, please follow the club, come back, and thank you so much. We are, you gave us an amazing talk and answered so many questions. We are very thankful.
Thank you, Katerina. Thank you, everyone. And yes, Gray Monroe and Detlef Weigel are good colleagues of mine, and they're doing amazing work. So I urge everyone here to attend their talk as well.
Yeah, great. Thank you so much again for coming, especially on such a sad day of the political and personal events that are affecting a lot of people and lives. I wish you all the best, and and I'll let me share again the link if you want to learn more about the current situation and or donate for humanitarian cause related to the current war in Russia. Here's a link. I have this link from a previous room that was held by by a journalist from NPR, National Public Radio. As far as I can tell, it's trustworthy, the links in there. So please go ahead and check them out. And thank you again. Enjoy. Have a good evening, nevertheless. And see you again tomorrow. Okay, thank you, everyone. Really appreciate it. Have a good evening, everyone. Bye.
Yeah,
Bye,
thank you,
everyone.
Katerina, for hosting the rooms. Bye.
Yeah, thanks, Kat.
Thank you. Thank you for coming.